Hello again and welcome to the NAFLDsym version 2A release webinar. Again, we appreciate everyone attending today. My name is Brett Howell and I'm president of DILIsym Services. I now have the pleasure of introducing our speaker today, Dr. Scott Seiler. He is our Chief Scientific Officer and has been a key leader for the company over the last many years, as well as our key scientific architect and leader. So he actually, a little bit of background, he did his undergraduate work at UC Santa Cruz, getting an undergraduate degree in biochemistry, and then did his graduate work at California, UC Cal, getting a PhD in nutrition. So with that, I will turn it over to Scott to take it away.
Thank you, Brett. And now I'm compelled to say, go banana slugs, the mascot of the UC Santa Cruz. In now addressing the topic at hand, I just want to kind of point out that it's really exciting to share the great work we've been doing with our quantitative systems pharmacology model, NAFLDsym. We had developed a first version that was made available for services projects over the last couple of years, and with support and efforts of many, we're now taking it a step further with NAFLDsym v2A. So as we transition to the following slides, this is our disclaimer. I'll let you read it at your leisure. So just by way of overview here on slide 3, let's just kind of talk about the problem and how we see NAFLDsym applying it or helping. First of all, NAFLD, or non-alcoholic fatty liver disease, with a subset of that being non-alcoholic steatohepatitis, or NASH, is thought to be a progressive disease of the liver, whereby the lipid burden placed upon the liver leads to hepatocellular injury, ultimately fibrosis and potentially cirrhosis, hepatocellular carcinoma, and a number of kind of associations with mortality as the disease progresses. The incidence of NAFLD and NASH is substantial and worldwide, much like as is the case with many metabolic diseases. At the present time, there are no real treatments aside from diet and exercise for patients with NAFLD and NASH. So this presents a substantial opportunity to help a vast number of patients worldwide as we collectively as a community are able to develop medicine to help treat these patients. There is a tremendous amount of resource being applied within the pharmaceutical industry to doing exactly this. And it's one of those situations where efficiency is really the name of the game. We need to get the medicine to the patients as soon as possible. And that is almost exactly where kind of one of the perfect spots for modeling to help with drug development. So it's with that in mind that we developed NAFLDsym, again, our quantitative systems pharmacology model of NAFLD and NASH. We'll go into this in great detail here, but the version 2A includes the primary components of the pathophysiology, including steatosis, lipotoxicity, inflammation, and fibrosis. We will be making this model available for licensing in Q2 of this year. And another really important feature, in addition to having kind of all the aspects of the pathophysiology, is the heterogeneity within the population. And we do capture that using what we call SimPops, or a simulated population. There are myriad ways that NAFLDsym can be used to support drug development for non-alcoholic fatty liver disease. Certainly, it's kind of combining the exposure, the pharmacodynamics, as well as the pathophysiology in kind of a unique way to predict the efficacy of novel treatments. It is a flexible framework that allows us to represent targets and treatments of a wide variety, which is exactly what is basically happening within industry at the present time. And really, it helps optimize clinical trial protocols in addition to predicting responses for specific treatments. And another, I think, really important aspect here is that, we don't believe at this point, the community doesn't believe that monotherapy, a single treatment, is probably going to be the real panacea. Rather, it's going to be polypharma. And from a mechanistic point of view, evaluating combinations of drug A with drug B that have different mechanisms of action is really a valuable resource. And NAFLDsym is well suited to do that. We've used it already in collaborative research agreements with several pharma companies. We've presented publicly some of the work that we've done in the past with Pfizer and Gilead, and Bristol Myers Squibb is presently sponsoring some of the development activity that we're sharing with you today. So before we dive into NAFLDsym in any great extent, let me just mention a couple of things about who we are, Deliverables Services. So we perform quantitative systems pharmacology and quantitative systems toxicology modeling. You can see that we have several platforms at various stages of development. Today we're talking about NAFLDsym. Perhaps our most mature platform is DILIsym, representing drug-induced liver injury. And then kind of more towards the infancy are RENAsym for drug-induced kidney injury, and IPF or idiopathic fibrosis sim, which will be available in years to come. But at the present time, DILIsym, NAFLDsym are available for licensing as well as services projects. And really, our vision, as stated in the big bold text, is to use modeling and simulation to really produce safer and more effective medicines. DILIsym Services joined the Simulations Plus family about 2 years ago now. And one of the things that really excited us about doing so is almost the perfect scientific complementarity. Certainly, Simulations Plus brings to the table, gold standard PBPK modeling capabilities. So the ability to predict the concentration of a drug at the site of the target. And so combining that with our quantitative systems pharmacology and toxicology models allow us to really even more poignantly make accurate predictions of response to drugs. So during the next hour or so, we'll discuss, I'll introduce you to the modeling software, NAFLDsym. I'll give you a brief demonstration of some of the features, as well as a snippet, shall we say, of an application with NAFLDsym, something that we've presented previously, and then just discuss some of the kind of the nuts and bolts, if you will, about licensing and servicing projects with NAFLDsym. So before we really dive into the components of NAFLDsym, one thing I found, and I think I've just now reached 20 years of the quantitative systems pharmacology modeling. And what I found over this period of time is it seems that everybody kind of has a different definition when they use that term. And so really to kind of make the lexicon comparable and make sure that we're all on the same page, I find it's useful to take a minute or two to do so. So really for QSP modeling, I think the focus is that the foundation of the model is a mechanistic mathematical representation of the pathophysiology. So what are the real key components of the disease? In this case, of course, NAFLD and NASH, and then capturing those mathematically. So that in doing so, you represent simulated patients, simulated populations. Of course, now we want to then see how they respond to drugs, and to do so, the next kind of critical aspect is to be able to predict the exposure, the concentration of the drug, again, at the site of the target. And this is where GastroPlus and PBPK modeling become extremely valuable, because some of obviously the targets are intracellular, some of them extracellular, circulating, all of the above. And then finally, combining the exposure, the concentration of the drug with the actions, i.e. the pharmacodynamics, the actions on its target, allow us to then determine at a certain dosing the magnitude of target engagement, and then superimpose all of that upon the mechanism and the pathophysiology to see if the drug is able to truly adjust and ameliorate, predicted to treat the pathophysiology. And it's of course at the intersection of that Venn diagram where we're able to then uniquely, I would say, predict efficacy with NAFLDsym. So the kind of the overview diagram of NAFLDsym is here, and it's a little color, it's somewhat color coded. You can see in the nodes listed in white are really those that are related to kind of the steatosis pathways. Those listed in red are related to the lipotoxicity. Those listed in blue are related to the inflammation, and in green is the fibrosis submodel. And So really, you can see in addition to those specific submodels, we also have a number of biomarkers. We'll describe those subsequently, as well as it's again important to note that we're able to simulate weight gain, weight loss, as well as kind of weight stable patients, because really it's a wide swath that we have, and we have to pay attention to how those factors can affect treatment. In order to build this model, we've relied exclusively upon publicly available data. There's actually a real nice collection of clinical data in the literature, and we've used those to establish the quantitative relationships to do the calibration validation, if you will. And then, as I'll describe here subsequently, we're accounting for the clinical and pathophysiologic heterogeneity with our SimPops. And of course, it allows us to then, putting all this together, predict the ability to predict responses to treatment. Again, not just monotherapy, but of course, combination therapies of all sorts as well. So with respect to the pathophysiologic variability and our approach for generating and representing simulated populations. So again, the approach we use is consistent with that used in most quantitative systems pharmacology, kind of standard within the discipline. And so we look at the disease and what is represented, in this case, again, NAFLD and NASH, and we identify key parameters that we know, A, play an important role in the pathophysiology, and B, where there are available data to define the range of parameter variability for a given aspect of the pathophysiology. So upon doing that, we apply the combinatorics of each parameter within the range and develop a large collection of potential simulated patients. Really what we end up getting is kind of interesting. So we have a vast amount of pathophysiologic variability, but oftentimes they kind of cluster into kind of common groups of clinical presentation. So F0, F1, fibrosis stage patients, those with steatosis, those without. And so we are able to capture, as well as the pathophysiologic heterogeneity, some of the clinical heterogeneity as well. These patients are subsequently, kind of more extensively evaluated and validated by comparing to clinical data. Initially, the clinical data that really capture kind of all the different axes of the pathophysiology. And then the next step is to, as well as possible, validate that they're giving appropriate responses to treatment. Again, at this point, the only available treatment for the patients is dietary intervention, so weight loss. And so all of the simulated population is validated against their response to a weight loss protocol. So let me take a couple of minutes to kind of walk through a couple of snippets of the data and how well the model is able to capture them throughout the four key submodels. So we'll start with the steatosis and lipotoxicity aspects. And so here, let me just orient you. So kind of consistently, our simulated population or simulated patients will be represented in red, whereas the clinical data will be represented in black, or in this case, kind of the dark gray. So if we turn our attention to the graph at the top, where you can see the x-axis is fasting de novo lipogenesis, or DNL, and the y-axis is liver fat. This is a study done actually by an old colleague of mine. She was a postdoc when I was a graduate student way back when at UC Berkeley, go Bears, and she went on to do studies such as this, which are a fantastic set of data that really nicely describe that patients with steatosis, NASH patients, tend to have elevated de novo lipogenesis. So fantastic insight, also useful to us as modelers in that it begins to establish what is the range, of de novo lipogenesis in patients with steatosis. And so you can see we did a wide sweep of the parameter range and came up with patients that filled every nook and cranny of that parameter range, as well as some that extended a bit beyond. And that allows us to do some exploratory modeling with some of those patients. On the bottom is the relationship between liver fat and ALT, which allows us to kind of get a sense of where lipotoxicity begins to manifest relative to the presence of liver fat, i.e., steatosis and lipotoxicity relationships. And again, in both data sets, you can see that the SimPops does a great job of representing, capturing really virtually the whole range to varying degrees. There are many other aspects. Again, we're kind of parsing it down just in the interest of time. So we'll move on to the inflammation submodel. One of the critical components, I suppose there are two. One is the number of potentially inflammatory cells, and two is the mediators that they produce. So this, in slide 13, illustrates, again, in red, how the simulated population compares with the measured data for lobular macrophages on the top and neutrophils on the bottom, as reported by Tajiri et al. In 2009. And so again, you can see that our simulated patients, categorized similarly based on the NASH score, really line up quite nicely with the clinical data, and allows us again to have some confidence that we're capturing the number of cells in the pathophysiologic state, capturing that appropriately with our simulated population. So moving from the cells to the mediators, one critical mediator is TGF-beta. Of course, this is the mediator that contributes to activating the stellate cells that leads ultimately to fibrosis. And you can see that using various data sets that we do, again, a nice job of capturing the range of TGF-beta across kind of the clinical spectrum, using ALT as sort of the indicator of severity. On the bottom is another mediator, PDGF, which helps to stimulate the proliferation of stellate cells. There's a kind of a dearth of data for this particular mediator. We were able to kind of extract a small set of data, and again, reasonably we're comparing to it, but acknowledging the fact that it's a relatively modest number of patients at this point in the clinical data set. And so of course we continue that practice, a number of mediators, a number of different aspects of the lipotoxicity and the steatosis pathways, and then carrying it forward to the fibrosis pathways, starting with the stellate cells. This is an interesting area for those of you who are quite familiar with NAFLD and NASH. The actual diagnosis or staging of fibrosis is dependent upon is as dependent on the location of collagen as it is the amount of collagen. And really, so then it starts with the activation of stellate cells in specific regions of the hepatic acinus. And so you can see there's the centrolobular, the periportal, and the midlobular. Collagen present only in the centrolobular gives you fibrosis stage 1, centrolobular periportal stage 2, all three, including the bridging then, gives you stage 3 fibrosis. And again, each of those are linked to the activation of stellate cells in those respective stages. I think, again, these figures nicely illustrate how the simulated population is capturing the full range of the data, as well as the unique aspect of the pathophysiology. So moving now from stellate cells to the amount of collagen, this is a in vivo, in patients study using isotopic tracers to measure the actual rate of synthesis of collagen, in this case, the type 1, what you see on the top. And you can see that, again, categorizing the patients based on fibrosis score with the measured data, that the simulated population is in excellent agreement with the measured data. Unfortunately, there wasn't the same for the collagen subtype 3, but we're illustrating the simulated population just the same, and obviously shows a comparable behavior. So putting together the synthesis rate with the number of stellate cells, ultimately we're also able to predict the amount of collagen. And again, categorizing it by fibrosis stage, using a variety of data sets listed down at the bottom of slide 18. And you can see that, again, we do a nice job of capturing the range of the amount of collagen for a given level of fibrosis. Another important category or aspect of NAFLDsym is the outputs. Obviously, you've seen a number of them now. In addition to those, and really any equation can be used as an output within NAFLDsym. But there are some specific biomarkers that are used to evaluate patients in preclinical and clinical studies. And again, in adhering to kind of classic QSP modeling principles, we've incorporated these outputs within NAFLDsym as well to align clinical measurement with predicted results. You can see in the blue are the plasma biomarkers, the red the histology measurements, and the black are the imaging measurements. So starting left to right to kind of highlight a few, obviously liver fat using MRI, liver stiffness using MRE, a measure of collagen amount, the histology looking at the steatosis, the NASH score, the fibrosis stage, as well as classic plasma biomarkers, perhaps the true classic being plasma ALT, but also some emerging ones such as plasma ProC3. In fact, I believe on the next slide, yes, is our representation, kind of a description of how our representation of ProC3 in NAFLDsym lines up nicely with the measured data. Now, ProC3 is something of an emerging biomarker of collagen synthesis. I think at this point in time when we're giving this seminar today, there's actually relatively limited amount of publicly available data. My guess is in the next 18 to 24 months, there will be a lot more. This is, I think, a commonly used biomarker at the present time, and I think there's a number of papers in the queue. And so we're excited to continue to kind of represent this using the ever-expanding amount of information. One limitation of ProC3 is it captures collagen synthesis irrespective of the location. So you have to kind of be careful with that. Nonetheless, it is really quite useful as a biomarker, certainly in evaluating treatment, not just pathophysiologic state as is given here. Moving on to a nice illustration of a specific imaging technique. So this is using magnetic resonance elastography, or MRE, to measure liver stiffness. Again, it gives you an index of the extracellular matrix proteins and the amount, the extent of fibrosis within a liver. And you can see the range using, kind of in the black and the clinical data from a number of studies, and how the SimPops really captures the range of response really quite nicely for a given stage of fibrosis. So now hopefully I've given you a snippet of how, again, we compare reasonably well to the various aspects of the pathophysiology in as much as it's described in the clinical literature. Now let's return back to the slide that we looked at earlier and highlighted in blue, and the response to dietary intervention in as much as that is the current kind of standard of care therapeutically. We've done a number of simulations, but I think this one nicely kind of summarizes the response. And this is really an emergent behavior. We weren't trying to tune or calibrate the response necessarily. And so really what you see here is a couple of studies where they had approximately 5% weight loss over various periods of time and used histology to evaluate not only the change in the NASH score, but also the various components of the NASH score, ballooning, inflammation, and steatosis. And again, you can see that in our simulated population, we do a nice job of capturing, with a fair amount of variability, which is often seen in these studies, the NASH score and these ballooning, inflammation, steatosis across the board. So again, what this allows us to do is have some confidence that the patients respond appropriately to treatment, but also it's important to note that there are a number of clinical studies where patients lose weight because they decide to take charge of their health, and of course that conflates the response to the drug potentially. So using this potential aspect of NAFLDsym, we are able to use simulations then to delineate the efficacy due to change in body weight versus efficacy due to the actions of the actual drug. And so that's an important component because, of course, sometimes you have very enthusiastic patients in a cohort, other times you don't, and nonetheless, you want to be able to have a true index of the propensity for your drug to help these patients. Okay, so throughout each of these slides, we've shown you really kind of the entirety of the simulated population, which is pretty substantial, approximately 1,500 plus or minus. But in reality, we don't anticipate that all users of the NAFLDsym software will use the entire population of 1,500 or so for every simulation. So as such, we have generated what we call sim cohorts, or subsets of the simulated population. We've developed a number of them that are based on kind of clinical categories that often patients categorized in this way. But perhaps more importantly is the software provides the ability to select specific patients from the larger group to generate a specific sim cohort that allows prospective clinical studies to be simulated in accordance with the characteristics of the anticipated cohort of that clinical study. This could be obviously done retrospectively as well, in past studies, and then select patients from the larger group that allows you to align nicely with what was done. Either way works, but the fact that the software makes it so easy to do is really an important point. And actually, it's at this point that I think is the appropriate time to move into a demonstration of the software. But before we do, I believe Brett has a question to ask of you.
Yes, thank you, Scott. We want to take a quick moment to engage you with a poll. So in just a couple of minutes, I'll be putting up a poll question. There are a few different options for your answer. Please, if you're inclined to do so, please enter an answer and then wait just a few seconds, and then the poll will automatically disappear for you. So I'll launch the poll now. So do you typically incorporate modeling and simulation solutions into your drug development programs? Going to wait for about 30 to 40 seconds.
We will reveal the outcome of this poll during the question and answer session at the end of the seminar.
Yes. Okay, so we'll give it about 5 or 10 more seconds. We still have some folks responding to the poll question. Okay, well, thank you very much for your responses. We're going to go back to our regularly scheduled program. Back to you, Scott.
Great, thank you, Brett. And thank you for those who submitted your answers to the poll. So I've obviously switched screens on you to now move into allowing us to have a bit of a demonstration of the software itself. So what you see in front of you is the home screen, and really it should be organized in a way, for those of you familiar with QSP modeling, in a pretty familiar way. So you can see the various parameter sets are listed here on the left-hand side. So there are species, there are drugs. So this captures the pathophysiology of the patients. The drug captures the aspects, the pharmacodynamics and potentially pharmacokinetics of the drug. There's caloric intake, allows us to represent, as we just discussed, weight loss, weight gain. You can obviously adjust the time to set different durations of simulations. You can adjust the solver if you like. And then another important point is down here on the bottom, we have a number of buttons. Obviously, you can run any given sim single, or you can run multiple sim singles in parallel to compare them. You can use this specific sim single to run in our simulated population or the subsets, the sim cohorts. You can do a parameter sweep, compare against data, and then view the various results in a variety of ways. So just kind of returning back to the species parameter set, I think it's nice to see an illustration of how the software really nicely summarizes each of the specific parameters that were adjusted to represent this specific simulated patient, number 323. So you can see the body mass, mild, pre-diabetic, various changes to oxidative stress and antioxidant capacity, the release of free fatty acids, and there are many other parameters as we scroll down as well. All of them summarized, viewable, and changeable should you choose to do so. So I'd like to, again, since I touted it so much in the last slide, I'd like to just show you real how easy it is to create a SIM cohort from the larger group. And so just by clicking this button and then kind of step by step, this window pops up, you're allowed to then, or kind of the next step is to select kind of the source, in this case, the NASH SimPops v2A. And then you would then select specific patients. I'll type in several here. Of course, I already did my homework, so I know which patients I think would be useful. And so you type them in larger groups or, kind of small subsets like this. So there are 5 patients here. You click on the Create Sim Cohorts button. And of course, you have to give it a name. Let's see, we'll give it in this name, if I can type properly. And save. Oh, okay, I guess we won't save that one because I made it for you already. So that said, it's really that easy. And in fact, I made it for you already so that we could use it to view some specific results. You'll notice again at the top of the SimSingle or the home screen, a number of different ways to access previously stored results. I'll click on the blue folder, which retains the results, provides access to results from previous runs with SimPops. And so I am now loading up the results with that n equals 5 sim cohorts in a theoretical ACC inhibitor. This is the parameters related to this specific compound are kind of made up, and so do not link to anyone that's currently in development. That said, so we ran it for 48 hours. You can see now the drug window is populated with the ACC theoretical parameter set. And then, of course, to view those results, clicking on the plot button, there are many, as I said earlier, many outputs. We use groups and subgroups to help kind of track and make it a little easier to find which you need. So for this case, why don't we start with clinical measures, circulating metrics, and we'll start looking at plasma glucose. It's a pretty standard one with which most people are familiar. And it also allows you to see how for each of the given patients, 3 meals per day are administered, followed by the subsequent excursion in plasma glucose. So we'll reset the panel and come back and look at total fatty acids. So these are the non-esterified fatty acids in the circulation. Again, adding that to the window, and you can see the same sort of thing where in the fasted state, they're at their peak, if you will, and then reach their nadir with each postprandial excursion due to the effects of insulin to inhibit lipolysis. Some other useful parameters to view, since acetyl-CoA carboxylase inhibitors are potent inhibitors of de novo lipogenesis. Let's look at that to just kind of confirm that we have target engagement, if you will, that the compound is doing as expected. So you'll see I went to the mitochondrial group, the lipotoxicity subgroup, and now I'm scrolling down until I reach liver average de novo lipogenesis rate. So I'll add that to the window, and you can see, starting at time 0, there's your normal de novo lipogenesis, and then in the presence of the drug, how it declines. It's also kind of nice to illustrate some of these features. If you wanted to look at the responses and highlight of specific individuals, you can do that. You can also export the plot so that it stays on the window, if that's your preference, or you can stay within this window. Finally, 2 other, I think, important outputs to view are the percent liver fat. And so you can see that even in the 2 days of treatment, there's a subtle decline in each of the patients. It's also worth noting, now that we're viewing this, that we actually have some patients that do not have clinical steatosis, right? So it allows you to capture for a phase 1, maybe single dose trial, you're able to use some patients that might be participating in that. And then finally, the last that I'd like to show you is the stage of fibrosis. And as expected, this isn't changing over the period of the 2 days, but it describes how in this small n equals 5 cohort, we have at least some patients that have fibrosis stage 3, 2, 1, and even 0. One other important note is that we've really taken great care to document the modeling work that we've done and provide that to users. And so you'll be able to see and have access to everything shown in this folder, the design notes, the references, the user manual, some descriptions of the SimPops, as well as spreadsheets summarizing all the parameters that are used. And then we also provide written documentation. Really kind of this, I would describe this as the modeling rationale. And so you can see here is kind of the overview of the entire model, and I'll actually scroll up and show you how in the table of contents we have each individual submodel described as well. So all of that is contained within the delivery of the NAFLDsym v2A software. So now at this point, let's return back to the slides where I'll take a couple of minutes to walk you through an application that we did. And this was a collaboration with Gilead, and we presented it most recently at a poster at the liver meeting in November in San Francisco. It was received quite nicely, actually. Speaking of ACC inhibitors, Gilead is developing one. And kind of the upshot of our collaboration is that the early clinical results of this compound seemed to indicate that the mechanism of action was more complex than initially believed. I'll describe that a little bit more here in a minute. And so generally speaking, this is a great way, to apply modeling to evaluate different hypotheses about mechanisms of action to see if they're consistent with what's actually was observed in the patients. This project was done in parallel with a phase 2 clinical trial. So basically both concluded at the same time. So we were able to compare the predicted responses with the unblinded responses from the phase 2. And it really actually lined up quite nicely, which allowed us to really hone in on and learn about these specific hypotheses. Also, coincidentally, Merck had around that same time published a paper describing the response they saw with their own ACC inhibitor. And so we use that as validation, again, really strengthening everything. So in the end, we were able to identify some of these key pathways that were activated by downstream gene expression effects. And this sort of insight provided from the modeling actually helped influence the clinical development program. So let me describe that here on the next slide. So this is a liver-targeted ACC inhibitor that has substantial and direct pharmacologic effects on de novo lipogenesis. They did a series of studies, first kind of a single dose target engagement, then a 12-week proof of concept study in a relatively small small cohort, then the phase 2 study. And consistently as they moved along, what they saw was that the compound was really effective at reducing liver fat, steatosis, but in some patients, there were also increases in plasma triglycerides. And that was sort of the surprising, if you will, result. And that's when they engaged us to really, help understand what might be driving that response. In the end, again, putting together all the information they've collected, including the modeling results, they were able to determine that co-administration of fibrates appears to offset some of those changes that may manifest as increases in plasma triglycerides, allowing them to continue development of this compound. So this is a very busy slide, I realize. And so just kind of highlight in blue are the direct actions of an ACC inhibitor. When you allow fatty acids to enter the mitochondria and you decrease the synthesis of fatty acids, we know that those are the direct pharmacologic actions. Based on some preclinical data, we had some suspicions that, due to this kind of this downstream effects of SRABP, that there may be as well some increases in VLDL triglyceride secretion, as well as some specific enzymes released by the liver that interfered with lipoprotein lipase, thereby reducing plasma triglyceride clearance. So again, we use NAFLDsym to simulate not only the direct actions of pharmacologic actions on de novo lipogenesis and mitochondrial entry, but as well as these specific changes in VLDL triglyceride secretion and plasma triglyceride clearance to varying degrees and varying combinations. And again, comparing to all the various clinical studies that we described a moment ago, ultimately simulating that phase 2 trial and comparing against the two as a pretty extensive table here. What you'll note as you review it is that surprisingly, almost, really quite nicely, the predicted response in red really lined up quite nicely, baseline and post-treatment across the board, especially at the 20 milligram dose, but really quite nicely for the 5 milligram dose, including not only the reductions in steatosis, but also increases in plasma triglycerides. Again, this really, I'll just kind of build this out, really influenced or, contributed to decisions that, where Gilead really felt like they were in a safe place by co-administering the fenofibrates, again, to ameliorate the potential increases in plasma triglycerides in those specific patients that really seem to have increases. So, in this way, working together and as well validating it using the Merck compound, not shown here, we're able to kind of allow this program at Gilead support their decision making, and it's continuing to be developed even today. So we have one more poll question, and then I'll conclude by just describing licensing and servicing project options. Go ahead, Brett.
All right, thank you, Scott. Again, I'll launch the poll. It'll be about 30 seconds, so please make your selections quickly if you'd like to address the question. Which of the following applications do you believe NAFLDsym is most applicable to within drug development? NAFLD-NASH target and or compound prioritization, or clinical trial design, or differentiation from a competitor's compound, or the evaluation of multi-mechanism combination treatments. Finally, all of the above. We'll leave the poll open for about another 10 or 15 seconds. Okay, go ahead and close it. Thank you for those who participated, and back to you, Scott.
Okay, thank you. So we'll conclude, as I mentioned, by just describing how you can gain access to NAFLDsym. First off, one is by engaging DILIsym services to perform services projects, and these options aren't mutually exclusive. We can do all of the above as well. But in this case, for our services projects, we prefer to do them in a staged way to allow the project sponsors to be apprised of the progress. And maybe there's new data that needs to be added and factored in. All of that happens in real time so that everybody's aware. Hopefully, I've given you a sense in describing NAFLDsym about how it's very flexible and captures, kind of the whole swath of the pathophysiology. Hence, is able to, we believe we're able to represent most all targets and/or compounds. But we'll have to discuss kind of project costs based on the required resources. There isn't kind of a one size fits all because, some targets, some are really well understood, some less so. So that's the sort of thing where, we would ask that you engage us. We'll have some early discussions to understand where you are with your program, what you understand about it, and then we'll, kind of begin to formulate estimates of the resources, hence the cost. So that's on one side. And now, really understanding that modelers really feel limited by the available options. They have tools, models to use. We're making NAFLDsym v2A available to QSP modelers via a licensing program described here. So for one instance, NAFLDsym v2A is $67,500. And that includes all of what you've just described, what I've described during this last hour, as well as some limited training, the documentation, and another really important aspect here, in addition to being able to add additional licenses at kind of reduced volume pricing, is that the equations are all viewable and modifiable by those who license NAFLDsym v2A. This is an important point because, just as I pointed out, everybody's working on a wide variety of targets, and so there has to be some customizability for the modeling community. So again, this licensing program can be done in conjunction with a services project or as an alternative, depending on the priorities within a given company. It's also really important to mention that in everything we described today, NAFLDsym v2A is really the efforts of a fantastic team, a great group of individuals, modelers from various and diverse backgrounds. And it's really their collective effort that allows us to be able to offer this to you here. And then finally, I'll just conclude kind of with the summary statement that we began with, which is really that NAFLDsym is designed to support drug development by predicting efficacy. So with that, I'll thank you for your attention, and we can open the floor for question and answer.
Okay, thank you, Scott, for the presentation. We do have a number of questions, so let's jump right into those, and I'll give the poll response answers in just a few moments. So the first question, does licensing of NAFLDsym allow for simulations on clusters?
So I neglected to mention the detail that at the present time, NAFLDsym is built on a MATLAB platform. So, you know, we have to kind of navigate the licenses of MATLAB. We anticipate future versions, by the way, being on more flexible and perhaps like a C++ sort of platform. But at the present time, NAFLDsym v2A is on a MATLAB platform. And so long as your cluster is consistent with MATLAB, there are some ways that, and we've worked with our clients in the past to be able to utilize their clusters. We believe that's also possible going forward. I mean, possible going forward.
Okay. Can NAFLDsym simulate disease intervention with a new candidate based on a new potential mechanism of action? And is it easy to add a new mode of action to NAFLDsym by a customer or through consulting?
Yeah, it's a great question. We're always learning about this particular disease state. It's relatively, despite clinically understanding its presence, I think we're really learning the pathophysiology now. And so the answer to that question is it really depends on the target. It really depends on the pathophysiology. If you understand it well enough, if the data are there to describe it, it is relatively low-hanging fruit. There are ways to, you know, certainly with experienced modeling practices, to represent some of the more complex ones as well. But again, that's the sort of thing I think we could evaluate together and determine whether, you know, in-house licensing and simulation versus a services project versus a combination of the two might be the best to meet your needs.
Okay, thank you, Scott. Can NAFLDsym be used for translational modeling from animal NAFLD-NASH model data to predict human responses? And how have you linked plasma biomarkers with histology outputs? Really two different questions there.
Okay, so for the first question, you were probably inferring correctly that we do not, at the present time, represent any preclinical models of NAFLD or NASH. So to do that sort of translational modeling, I think we would work together to really understand the preclinical data you have in hand, as well as, just as importantly, which preclinical model you are using. As I'm sure you're well aware, each preclinical model is a little bit different than another, and it seems most companies have kind of a unique preference, which is one of the reasons why we haven't represented them. Nonetheless, we would understand, you know, what you're seeing in that given preclinical experimental environment, and then determine the best way to translate it. Obviously, there would be some uncertainty about that. We would potentially do some sensitivity analyses to really understand the parameters, and then guide. Really, I think that in those sorts of situations, guide data collection and subsequent experimentation. For the second question, linking plasma biomarkers to histological measurements, some of the obvious ones are, for instance, ALT, CK18, and the ballooning histologic measurement. You know, ballooning is representative or an indication of cells that have expired due to apoptosis, and those biomarkers can give you an index of the rate of cell loss, and so that's an obvious one. For inflammation, you know, obviously you saw there were a number of mediators that we simulate, circulating mediators, and so to some extent they provide some sort of an index there. And with respect to then the histologic measurements of fibrosis, really that probably lines up best with the imaging, something like an MRE. But there may be other plasma biomarkers that also line up. This is a really rich and field in terms of the attention it receives, the biomarkers of NAFLD-NASH, with, I think, also, I want to say ambivalence. I think everybody seems to favor a specific biomarker, but the community as a whole hasn't landed on kind of the classic indicator like an HbA1c would be for type 2 diabetes. So, we're trying to stay up to date as best as we can with that, but it is sort of a moving field.
Thank you. How much would protein expression of enzymes and transporters in NAFLD be taken into account in the NAFLDsym model? And if so, what's the rationale?
Also a great question. And I think it's at this point that it's important to delineate, especially with respect to transporters, those that are responsible for the uptake of compounds, kind of uptake of drugs, versus those that really participate in the pathophysiology. I think there's an ever-increasing amount of data that indicates that, certainly from a pharmacology point of view, drug transporters, the expression of each are really quite different. And so to capture that, I think that's where we really use something like a GastroPlus and really make some adjustments to parameters there to make sure that we're capturing those aspects of NAFLD and NASH. There are other proteins, obviously, contributing to pathophysiology, for which expression is important. The example of the project with Gilead, where there were some clear changes kind of after the direct pharmacologic actions that allowed us to then understand some of the downstream and overall efficacy responses. And so having access to that information, preclinically or what have you, allows us to evaluate those mechanisms of action as well.
Okay, thank you, Scott. Can you say whether you've simulated ASK1 inhibition, and if so, how did that go?
Yeah, this is something of a hot topic at the present time, and so I think the appropriate way to say it is no, we cannot speak to that specifically, but we're certainly aware of the target and the compounds that are, some of the compounds that are in development at the present time.
Okay, another question. Can the slides be shared with the participants? I can answer that one, yes. We can certainly share the slides. Can you discuss at a high level the process to simulate a drug against a specific mechanism within the model? Is this programmatic?
So again, at a high level, if I return back to slide 7 and build it out, really we need kind of these three components to be able to do so, and really the pathophysiology already represented within NAFLDsym v2A. So what we need to do then, kind of the next step, is to be able to formulate and develop a PBPK model. Maybe you've already done so in-house and we're able to use that, or again, we can use GastroPlus and our Simulations Plus approaches to do so. Then the next important aspect is really understanding the target, which kind of downstream pathways are affected, in order to really assess the pharmacodynamics, i.e. the actions of the drug and how they interact with the pathophysiologic pathway. And so to do that, again, we need to know a bit about the studies that you've done, what data are available. In some cases, the target's really well understood, other cases not as much. But and then obviously we're able to do this, once we have all of the basic components captured via equations, then, really the unique aspects of each compound, be it PK aspects, be it PD aspects, allow us to evaluate compounds in a library and to determine, what might be the optimal compound and, versus those that maybe should really be relegated to a backup.
Thank you, Scott. Were the ACCI simulations prospective? For example, open the envelope for plasma triglycerides.
So the answer is a sort of, I suppose. We had access to the 12-week proof of concept N equals 10, N equals 12 cohort data. But the phase 2 clinical trial was occurring in real time as we were doing the simulations. And I mean, really the data were unblinded almost exactly on the same day as the simulations were completed. So actually, if nothing else, the data were unblinded after the simulations were completed. So it really was prospective in that way, again, influenced by that 12-week really small cohort study. So for the most part, yes, I would say it was unblinded, or sorry, prospective. Is that all the questions? It seems that I've lost Brett's audio.
Sorry.
Oh, there you are.
Back here, yeah, it apparently got muted. So there was time for one more quick question. You were simulating glucose response to feeding. How about meal triglycerides, for example, chylomicrons, or effects of meal protein, for example, gastric emptying?
Yeah, also a good question. So plasma triglycerides, chylomicron, VLDL, all represented within NAFLDsym, yes. The amino acids in the plasma protein, or sorry, in dietary protein, not as much. You know, largely because they're more passive participants in the steatosis process. That said, you know, kind of the emptying of meals at this point represents kind of a standard meal type, although it is modifiable. So yes, it is. We are able to capture some of those interesting dynamics of interesting meals if needed.
Okay, well, thank you, Scott. Just a quick note on the polls. For the first question of do you incorporate modeling and simulation, 59% said yes, 12% said no, and 29% said not applicable. And for the second question, which of the following applications do you see as most useful from NAFLDsym? 35% said NAFLD-NASH target or compound prioritization, 23% said clinical trial design, 19% said differentiation, 39% said evaluation of multiple mechanisms, and finally, 58% all of the above. So let's see, that's strange because I think there were multiple votes there by some folks, multiple different answers. But with that being said, I want to thank you, Scott, for your presentation, and thank you all for dialing in. Feel free to engage with us afterwards, and we'll be happy to answer additional questions and discuss NAFLDsym and how we can engage with your organization. So again, thank you all very much, and we'll wrap up the webinar.
Thank you.